In this video, we're going to look at the secretory pathway. Now, any protein that needs to be secreted or lives in the ER or is lysosomal or transmembrane will have to go through this pathway. So let's take a look at the steps of the secretory pathway. First, any peptide that needs to go through this pathway will have a 13 to 36 residue signal peptide. Here are a bunch of these signal peptides, and they're all similar in that you have 6 to 15 hydrophobic amino acids flanked by hydrophilic residues, and you always have a positively charged, well, not always, mostly have a positively charged residue near the N terminus. Now, the sequence of these signals are not similar, but they all form an alpha helix in a nonpolar environment. So it has the same sequence, or the same structure. So remember, structure is more important than sequence. Now, when the peptide is roughly 40 kilodaltons long, the signal recognition particle, SRP, will bind the peptide in the ribosome. And during this process, the SRP's GDP will be replaced by GTP. When this happens, you'll have a conformational change in the SRP, and that will stop the ribosome from making any more of the protein. Then this SRP ribosome complex will diffuse to the rough endoplasmic reticulum and bind the SRP receptor in complex with what's called the translocon. And the translocon is simply a protein pore in which the rest of the protein will be extruded. Now, the receptor has GDP, and when this binding event happens, that GDP is replaced by GTP as well. Now, when this docking happens, the SRP and the SR each hydrolyze their GTP to remake GDP. When this happens, you have another conformational change, and the SR and SRP will dissociate from each other. The ribosome is now free to continue making the protein. And the N-terminus will go through the translocon, and once it's through, the signal peptide is actually chopped off. So the signal peptide is not part of the protein. It's just a signal to say that this protein needs to go to the ER. Now, this protein will start to fold in the ER, and it will be helped by the chaperone protein HSP70. Now, inside the lumen of the rough endoplasmic reticulum, you have enzymes that will start any post-translational modifications, such as carbohydrates or disulfide bonds via disulfide isomerase, which we talked a few weeks back about. Now, once you're at this step, there's no turning back. You can't go through this pore again. It's a one-way channel. If you have a transmembrane protein, you'll have one or more 22 residue membrane anchor sequences that is basically a signal that this sequence needs to be in the membrane. Now, after synthesis, the ribosome and the translocon will dissociate, and the ribosome can fall off. And every single life form that we have ever looked at have similar proteins to everything that is mentioned here. So this pathway is seen in every form of life in some way or another. Now let's look at the translocon a little bit more. So translocons have an aqueous pore, that is the center, allows water to flow through, and they span the entire ER membrane. Now, they're a heterotrimeric protein, which means they have 3 subunits that are all different, so alpha, beta, and gamma. It's Sec61 in mammals, while in prokaryotes, the translocon is called Sec4. And a translocon is seen in every single kingdom of life. And these proteins have a lot of different alpha helixes, and the channel is not always open, but rather it's blocked at the extracellular end by a short hydrophilic helix. A signal that that will come in and say that a protein needs to go through the translocon basically moves this helix aside. So you can think of the helix as a plug that blocks one side of the translocon. A signal comes in that the translocon needs to be used. This plug moves aside. Now, translocons also help the insertion of any integral protein's transmembrane helix segments. And this happens at the C-shaped subunit. So this would be the alpha subunit. The signal peptides of many transmembrane proteins are actually not cleaved but inserted right into the membrane. And these are called signal anchor sequences. So we looked at a couple of slides back how the signal sequence is chopped off. This actually doesn't happen for transmembrane proteins. How the translocon inserts transmembrane sequences, we don't really know yet. We just know it is vital for that process. That's it for this video. I will see you in the next one.